can we try to manage marketable yield by our cultural practices? And you had a number of things you could do. But 2 on the list was no harvest and no smart fresh. So I would like to know the science behind that.
That comes from trials that we've done with Chris Watkins. Wherever we have high potassium to calcium ratios, and somebody already typed in what is the desired potassium to calcium ratios. This is based on peel sap analysis that Lai Liang Chang developed. And he and I have set these ratio limits. If you're below 23, things are relatively safe for long-term storage, and Smart Fresh and Harvistus and Retain seem to be okay. There's this intermediate range of potassium to calcium ratio between 23 and 27, where it's a little iffy. But if the ratio is above 27, we see problems with Retain and Harvista causing more post-harvest storage disorders, including bitter pit, but also senescent breakdown. So we are pushing in New York. We'd be happy if anybody else in the whole country could copy this or modify it, but trying to get every block sampled in early July for a fruit peel mineral analysis using peel sap that Lai Liang's developed, and then decide what to do with that fruit, whether or not you put Retain on it or not, or Smart or Harvista. Because if you have high potassium to calcium ratios, we see tremendous increase in post-harvest disorders. Harvista can be applied later, but still, we want to be able to react. And this timing that Lai Liang's picked with Mario seems to give us opportunities to do something that same year to try to manage that crop when we end up with high potassium to calcium ratios.
We are, for your information, Terence and Laila knows this already, that we are contributing to the project with a sampling this year. We want to evaluate how that indicator works in Washington. But for the meantime, for answering the questions that growers always have, do you have a value in terms of potassium to calcium ratio that they should follow in fruitlet?
Fruitlets, no. I think in general, no, not for fruitlets, but for full fruit, for peel, it's pretty close to what Lai Liang had developed. It was 25, above 25 to 1 is at higher risk. But that's for larger fruit closer to harvest.
Yeah, so I want to second what Terence said, because to my experience, to this my previous experience in Italy, we do 2 sampling. So one is in June early, and immediately after we end up the end thinning. We don't do a lot of end thinning, and then we do in July. It is exactly what Terence was telling you. In June you can have an idea if you are already out of range, and then you can correct eventually. Irrigation, nutrition, all this kind of stuff, and then we don't speak anymore. Otherwise, we spoil the next meeting. And in July, of course, you have another option to check where you are, and you still have a possibility, because if you go in August, everything is almost done and will be very difficult. Somehow you have to think about the spray, the calcium, the form of calcium that you are spraying. They are chelates or something like that. So it's a complicated issue.
We have done over years, we have done tons and tons of fruitlet analysis for early prediction. And in mid-1980s, we had several models that we published at the ASHS trying to predict the quality at harvest using either fruit, leaf, or combination of those, either early in the season, mid-season, late season. The problem when we go too early is that the total or the final crop that we have after thinning does not correspond very well with that very early analysis. It seems that after August or sometime mid-August, after the minimal flux of the mineral nutrition approaches, it means that the maximum amount of carbohydrate is in the fruit, they don't change too much. That's the time that minerals are stable. That's why we are recommending to growers to take their leaf samples at that time. It seems that what Terence suggested, which is an excellent one, I would push it a little later for, I guess for the Honeycrisp, that would be okay because it is harvested earlier than Fuji. But we want to make sure that minimal flux has been reached before we do that. If I make one more very quick comment, and that is Terence's presentation, he mentioned, and it is very much true, that we have to look at the combination of many things. For example, in the West, in Washington, Idaho, we worked on 25 years on the irrigation and nutrition part of that. It seems that no matter what you do, what rootstock you have, amount of irrigation by itself, if you have more irrigation, you have increased potassium in the tissue. Therefore, in your Honeycrisp, that would be a danger. Absolutely. Then you get more potassium to calcium ratio. Another thing is that more vigorous rootstocks, naturally, they have more potassium in them, but they have larger size too, more often. So we should find a happy medium somewhere. Thank you.
What is the availability of 214?
We should be at about 1 million trees now, or rootstocks. So it's coming up fairly strong, and it should be more and more available. There was one issue with 214 that impeded the quick establishment of a lot of stool beds. The micropropagation went very, very well, and you could basically make about 500,000 plants or little plantlets. However, when it went from the plantlet stage to the acclimation, there was maybe a 30% success rate. And so that made those little plantlets very, very expensive. And those plantlets were supposed to be used to establish new stool beds. I know some nurseries have paid up to $4 a piece to do that. And now they have, once it's in a stool bed and once it's there, it's a wonderful rooter and good, essentially very good rootstocks. But yeah, the problem with 214 has been the tissue the latter part of the tissue culture process.
Another rootstock that we haven't talked here is Geneva 4004. That has been an excellent rootstock for us. I should emphasize some of my experience is mostly on Fuji. So we really like that. Although it may be considered that as a tough, more vigorous than the other ones, but we like it because our soils are weak here. Most of the soils that we have here.
So thank you, Essie, for mentioning 4004. We're in the process of releasing it. It will be our next release, and we have virus-free wood and everything, and it's being deposited in the right places so that we can make a thing. But the fact that you mentioned 4004 working very good for you in Idaho, it basically proves my point of designing rootstocks. We need a diversity of rootstocks available to our growers, and sometimes it's going to be somewhat localized to a certain industry or a certain state or a certain type of soil or whatever. Having a few more available and that knowledge that people like you, Essie, are developing, that localized knowledge, is very precious. So 4004, it's in the process, as well as 257 and a couple more. So,
Tom, could you make a comment on the relative dwarfing effect of some of the Geneva rootstocks in your experience in Washington and versus the chart developed at Cornell by Terrence and Gennaro? I did notice some difference in terms of relative ranking.
I think one of the biggest differences is that our trials were all done at 10 by 3 or even some angle canopies down at 12 by 1.5 feet. And looking at a demonstration in New Zealand where they looked at planting density and the impact on productivity, it was very, very interesting to see that the higher density at 30 inches had tremendous yield advantage over the 36 or 60-inch spacing. The 60-inch spacing was just a disaster for wood caliper and that sort of thing. So when we look at how the Washington trials were managed, they were all reasonably high density and managed to keep the large wood that's not productive pruned out of them. And so we encouraged a number of those rootstocks to be very productive. There's two things that are very unique about many of the replant tolerant Geneva rootstocks is that they grow very vigorously non-bearing. They grow vigorously in the nursery, they grow vigorously in the orchard. However, when they crop, they can be calmed down very well. G890, we actually need to add fruit to it as soon as we possibly can in order to make sure that the portions of the tree that have a lot of growth will calm down and keep that wood size in a productive range. But as we crop these trees, particularly second, third leaf, the crop took the vigor out of the trees very, very quickly. And so the non-replant friendly rootstocks like Bud9, if we were on an unfumigated plot, we could crop those trees to death in 2 seasons. And so we lost a tremendous number of Bud9 trees just because of the cropping on those unfumigated plots. The other thing that's very interesting with replant and the vigor that we can maintain in the Geneva rootstocks is that we would get out to year 7, 8, 9 and see that the unfumigated plots of Geneva rootstocks were actually growing more and were more productive than the fumigated plots of M9 or Bud9 or any of the non-Geneva rootstocks. And those findings were kind of supported by Mark Mazzola, a plant pathologist at USDA Wenatchee that surveyed the microbial growth under these different rootstocks and saw that G41, for example, completely changed the microbial growth under the rootstock from the root exudates produced. And so that was a big factor in being able to sustain the vigor in a highly productive scenario. So I think one of the interesting things that we need to follow up with is how we design our production systems, multi-liter trees or even single trees. We need to find that good tree spacing that allows that canopy to be full for cropping as uniformly and as quickly as possible. So for like Bud9, you know, it's 18 inches to 2 feet apart is in the row spacing. 214, 41, maybe 30 inches, 36 inches. So the row spacing and the leader spacing on multi-leader systems will be very, very interesting to follow and see how easy Stefano can make it to maintain.
Some of the Geneva rootstocks, those replant disease tolerant Geneva rootstocks, develop more associations with beneficial microorganisms, less association with pathogenic microorganisms in the rhizosphere and also inside the root system.
Does anybody got a comment on irrigation relationship to rootstocks, relationship to bitter pit and deficit or not deficit irrigation?
I would like to follow up on that question, Phil, and And this is related to what Essie mentioned on irrigation to manage the bitter pit, but also I read from Gennaro, you published an article that I thought it was very interesting related to the hydraulic conductivity and ABA on the different rootstocks. Can you expand on that and talk about that resistant to drought or the opposite?
Oh goodness, that is a deep question. I was going to mention, I was going to go down to the into the hormone and phytohormone lane eventually, because I think someone asked about spraying Harvesta or other things, and whether rootstocks would have different reactions or there could be an interaction. And the quick answer to you is yes, rootstocks do produce, and this was measured in Honeycrisp in the xylem. They do change the hormonal status of the scion or of the grafted scion. So, it remains to be seen. So there are rootstocks that are very, very high in ABA. G11 is one of them. There are rootstocks that are fairly low in ABA. But then also there are differences in other phytohormones. The question about water use efficiency or drought tolerance is that it's a tough one, because you not only have to consider the rootstock and what it's the hormones, ABA, that's sending up to the scion, but you also have to consider how the scion is being managed, and that can have a huge effect on drought tolerance or water use efficiency. So it's a topic that I'm going to be studying in the next 5 years, and I will have better answers. Because if you take just the knowledge of the rootstock alone and say, oh, it produces ABA, therefore it closes the stomates faster or more than another rootstock, you're likely going to be missing the big interaction that's up top. And even pruning can have an effect on water use efficiency and the canopy development and so on and so forth. So and there was another study done by a French group that looked at the density of the stomates in a leaf, and the varieties change. So about that, and they also change with regards to sensitivity to ABA. So some shut down very quickly and keep that evapotranspiration down, and some they are not that sensitive to ABA. So this is going to be a topic that's going to take quite a bit of research.
Bernardita, one quick comment in what Gennaro mentioned. He mentioned about the role of variety on the rootstock. I know we are talking about Honeycrisp. I think our major growers here remember in 2014 that we had major damage in the West because of the November freeze, that we were 50 degrees Fahrenheit or 60. And then on the 17th of November, plunged down to -5. We had a vast number of trees in Fuji. They were severely damaged, and none of the rootstock next block to it in our research orchard, next to it was our Honeycrisp on different rootstocks. None of them died, none of them. And then we had V series, we had some Genevas. We were hoping that some of them come better than the other ones. In Fuji, we had damage in all, particularly on M9. But the next to it was the Honeycrisp came without any damage.
Mario, I'd like to try to answer 3 questions during the question and answer, and then pull Lee into this question on irrigation. Let me first answer a question on whether or not Apogee or Kudos applications at Pink help in terms of calming down vigorous rootstocks. It appears that yes, they do, but probably not so much for growth control. There's not really time for Lai Liang to tell all this theory, but the theory he has, which I tend to like a lot, is that it affects the cell division in the pedestal of the fruit, which allows more calcium into the fruit. So yes, it is a helpful thing, but it's not a solution to the vigor problem. It's a partial solution. Number 2, there's a question on NIC 29. Is there any information on it? Yes, we have information on it. It reacts very similar to PAJAM 2. There's really no difference in terms of the bitter pit with PAJAM 2 and NIC 29. Number 3, is pruning a way to keep trees within their space, or is it all the rootstock? It's both. The style of pruning we do on high density orchards, particularly when we switched to tall spindle and then super spindle, was to remove large diameter limbs. That in and of itself is a dwarfing process. But in addition, the rootstock provides a tremendous amount of dwarfing. There is some theory that the competition in very closely spaced trees between water and nutrients is a reason why they're smaller. I think there might be some of that, but it's lesser effect compared to what we're doing through pruning by taking out big branches. I've said this for years. Big branches make big trees. When there's no big branches in the tree, the trunk stays smaller. Our data from a 2005 to 2008 PhD study showed that drought in the early part of the season between bloom and 4 weeks after bloom gave more bitter pit. And I predicted that, I presume that's because we're limiting calcium uptake. But secondly, drought in mid-July also gave us more bitter pit. So those are the 2 typical times we can have drought here. We often in August get rains and we don't have the ability to control it. But certainly in my data, dry weather increases bitter pit, and I presume it's for lack of calcium uptake.
So I'm going to keep my comments in the context of rootstocks, but you know, we've done some work experiments looking at how different rootstocks respond to water limitations. Something like Bud 9, because it's already so dwarfing, you're not going to be able to increase the growth of Bud 9 by watering it more. It's really difficult to do that. I mean, it helps, but it's going to be a very small increase. But something like G890, which is a more vigorous rootstock, it's going to be highly responsive to how you water. We've seen that it increases the diameter of laterals, so you're changing the thickness of all that wood, increasing pruning by increasing water, and also just changing how that tree yields long term in terms of its biennial cycles. So if you're overwatering G890, it's highly responsive. So in irrigated areas, when you have 890 and you have high growing degree days, you're in a hot area, it's going to be a challenge because it's going to be difficult to control, but you can pull back on the water and change its responses. So G890 is much more responsive, and you're able to manipulate growth, manipulate fruiting sites, and manipulate branching using that. But you lose that control with Bud 9. Bud 9 is a great rootstock, but you don't, it's already naturally so dwarfed that you're not going to be able to change that very much. But some of the more vigorous rootstocks, and I think the comment about Nat cheese being up at 2,200 feet, it just has less growing degree days. It's a cooler site. It blooms a lot later, so G890 is better suited to that just for that reason.
Just a quick reminder that our next meetup is in 2 weeks, and that will be nutrient management, as we've said several times on this webinar. So if you haven't registered already, we've been sending out those updates, and I can put that in the chat window, but I think that most of you probably have. And thanks for joining us tonight.